Thank you for the invitation here. I was telling Mark and some others a few minutes ago that actually my very first scattering experiment was done at APS, not at your beamline, but at APS. I was an undergraduate. I was looking at inelastic X-ray scattering of this really weird material. It's a free electron liquid, and so I didn't end up going in that direction in the rest of my career so far. But it was a great experience. As an undergraduate, it was fantastic to go to one of these facilities, and I find it very funny, of course, that I'm now working at one of them at NIST. So this talk is about autonomous neutron reflectometry via information maximization. And I want to just start out by thanking Frank, who's on the call today, who really laid the groundwork for this with his work into in exploring what sort of information content is in a neutron reflectivity curve. And that really set us up. And he and I have been working closely on this project to talk about how do we maximize that information, either by planning the experiment, which is what he's really focused on, or kind of in real time, which is what this talk is about. I also want to thank my colleagues Brian Moranville and Paul Kinzel from NIST. They've built a lot of the software tools. Brian is really involved in the ORSO reflectometry organization as well. Paul wrote the REFL1D software that is a fitting software that's widely used for neutron and X-ray reflectivity data. And I'll talk a little bit more about that in a bit. All right, so let's, also a lot of this is funded by the Center for High Resolution Neutron Scattering. That's high resolution for neutrons, not high resolution the way you guys think of high resolution, but it's an NSF funded sort of consortium between NCNR, the NIST Center for Neutron Research, and the NSF. And it funds a lot of this sort of work that we do. So the inspiration for this really came from a workshop that Frank and I went to a couple of years ago called, it was a workshop for autonomous scientific experimentation. And I'm sure many of you are quite familiar with this. When you're thinking about planning an experiment, there are often really two big questions. First of all, what do I measure? What samples do I run? And then secondly, how do I actually go about collecting my data? And in both of these cases, often it's useful to have some sort of artificial intelligence or machine learning algorithm that helps make some of these decisions for a user or for a researcher. And just to give two examples of that, how do I collect my data? This is this was an experiment that was run at Brookhaven National Lab at one of their diffraction beam lines, and they were looking at this evaporated. This is like a coffee ring structure. It was evaporated solution of nanoparticles, and if you look at this paper, what they show is that they can efficiently collect data about this by this sort of active learning, the same sort of active learning protocols that I'll talk about today. And the reason that you want to do that, of course, is that there's a lot of this is empty space. It doesn't have any interesting information. So you want to be able to, in real time, sort of drill into the parts that are going to give you the most information. Similarly, in Spirit, if you want to actually discover new materials, this is one that came from the NIST Cameo group for this sort of tertiary material. A phase diagram actually looks a lot like an image in some ways. And so if you have the laboratory automation that can back you up, you can actually explore this phase space. Each one of these dots is a different material and try to optimize for whatever parameter you want. So again, very similar in Spirit. And of course, you can extend this to higher dimensionality than just 2 or 3 as well. I also wanted to give a shout out to an effort that Frank and I are leading at NIST called Roadmap, which is to actually do a very similar sort of thing with membrane active peptides. And this project is so new that we don't even have the website up yet, but give it a couple weeks and we'll probably have a website that you can take a look at for that. Yeah, we just got the funding for that a couple weeks ago. So what we're going to do in this, reflectivity, of course, is a one-dimensional technique. So we're actually going to reduce the dimensionality of the problem and go down to 1 dimension, and we'll focus a lot on collecting data. So as an example, of course, for this audience, I don't need to describe what reflectivity is, though there are some differences, of course, between the way you would do an experiment in the X-ray case and neutrons. So this is an example of a fairly straightforward soft matter sample that we would do reflectometry on at NIST. And this comes from a larger work with Dennis Michala, who was a graduate student at the time, and Matthias Loesche from Carnegie Mellon University. So in this case, we have, so this is not this is a solid liquid interface rather than a liquid air interface. We have a silicon wafer, it has a native oxide on it, a titanium oxide film is sputtered on it. And then we have, described in the paper, we have a technique for preparing lipid bilayers on top of this. And what we're interested in this particular case is the structure of this lipid bilayer on top of the titanium dioxide surface. And the way that we typically take measurements with neutrons is using this thing called contrast variation, because we're scattering from the nuclei instead of the electronic structure. We could do this trick with isotope exchange. So if we do the same experiment in D2O based buffers or H2O based buffers, we get two complementary data sets that gives us contrast between the various components of the system. Basically, it tells us where all the water is, and then we can kind of back out what everything else is from there. The data that you get from these two cases, shown here in this maroon and green, look quite different, of course. In the D2O case, you have a critical edge, for example. In the H2O case, you don't. And the other thing to remember about what I'm going to tell you about today is that because neutrons are, the sources tend not to, are many orders of magnitude less bright than X-ray sources, we tend to be statistics limited in what we're taking. So that is actually also an important caveat. Anyway, what we would do then is we would create a model in which we simultaneously fit both of these curves to our model and back out what these scattering light density profiles are. Now, actually, under the hood, what we usually do is something that that Frank and Matthias Loesche developed over many years, is this composition space modeling, in which we break the system up into its components that have sort of unique scattering light density. So in this case, we would say the silicon is one material, silicon oxide is another, titanium oxide is another, and then the bilayer itself is made up of inner head groups, outer head groups, and the lipid species. Each of these has basically a constant SLD. We can create distributions for each of these, which are constrained based on the known stoichiometries and volumes in the system. And then we fill in all the gaps. We replace those with various parameters in the model. We fill in all the gaps with water. And so basically, when we change a parameter, for example, the submembrane thickness, this whole assembly would move out away from the substrate or closer, for example. In this particular model, we have some parameters which we're interested in. So the physical parameters we're interested in are the submembrane distance of the bilayer from the surface, the inner and outer leaflet lipid thicknesses, the completeness of the bilayer, so how much water is actually in this bilayer, and its roughness or its fluctuations. And then there's a lot of other parameters in this model as well, and I'm sure you're all quite familiar with this. So things like the thickness of the silicon oxide, or the thickness of the titanium oxide, or the surface roughness of some of these things. There are 11, in this particular case, there are 11 other nuisance parameters in the problem that we really don't care about. So this is going to be an important theme of this talk, parameters of interest versus these sort of nuisance parameters. For an experiment like this, we can also talk about our levels of decision-making. And in this case, we might talk about whether we use titanium oxide or something else. So this is the kind of, how do I design my sample? Which lipids do I use? Do I use D2O and H2O, or do I use some mixture of them? And then there's another question in here too, which is particularly important, I think, for intensity limited techniques like neutrons. How much data do I actually need? When can I stop? Like, when do I have the data that I need? And this is one that we typically don't ask so much because it's difficult to make that decision in real time. And then finally, how do I collect my data? Which instrument configurations, what angles, what Q values do I measure at? And then how long do I measure at each one of those? So these are all questions, again, where we think that this might be a place for AI or machine learning, or in this particular case, active learning can play a big role. So to see how that might work, let's talk about how the data that I just showed you would typically be taken. So let me just go back and show you the data again. So these data are taken. You can see that each of the Q values is evenly spaced. Kind of at low Q, we have a tighter spacing, but each one of these Q values is evenly spaced. Because the reflectivity drops off as Q to the fourth, and we also open our collimation as we go to higher angles, to get a constant delta R over R, we typically say we should measure the measurement time per point should scale with Q squared. And we have this well-defined collimation as a function of Q that gives us these relationships. Okay, so this is something where we would define at the beginning something called a trajectory, where we say, these are the Q points I'm going to measure at. This is the amount of time I'm going to spend at each one of those Q points. This can be a complicated relationship, but it's all set at the beginning of the measurement. Then we go and we actually run this on the instrument. We collect the data, we reduce it, so subtract background, normalize the intensities or whatever, and then we go ahead and analyze it. And here again, we need some user input, where we have some model that I showed you previously, and some parameters of interest that we might identify that would then end up, for example, in a publication. So this is how we would typically take a measurement. This is with our monochromatic reflectometers, by the way. I'll talk a little bit about polychromatic beams later in the talk. Now, to do this in an autonomous way, the reason we'd want to do this in an autonomous way is to actually maximize the rate of information acquisition in the measurement. And the central assumption here is going to be that some regions of Q space are more efficient than others at constraining the parameters of interest. So hopefully this is sort of obvious to you in the sense that take some random reflectivity curve that has a critical edge. Below the critical edge, for example, you don't gain any information about your parameters of interest, about the structure of the interface. At really high Q, and this is particularly important for neutron measurements, your signal to noise degrades. So if you go to high enough Q, you're also not going to get much information. So you know in some sense that this is true, but then is it true enough to really make a difference? That's going to be the thing that we're going to explore in this talk. Can we identify some parts of Q space that really help us hone in on what we want to learn in the experiment? And we want to spend our time there. So how do we have to change this workflow to actually do that? Well, we add a loop. Now our set of instrument configurations is a set of possible measurement configurations. We collect some data, we reduce it, we analyze it, and then we have an algorithm that identifies our next data point to measure. And this is going to be informed in this talk anyway by our underlying model and the parameters of interest that we identify. And then we go collect some more data at the data points that we identify as we think being the most likely to give us the information we want. We reduce that, we analyze it, and we go around the circle. Okay, so this is how an autonomous measurement works. The identification of the next point, I'm going to call that algorithm. It's going to produce something called a figure of merit, a sort of optimization function, or in other parts of this field, it's called an acquisition function. And also I'll talk about how to make this specific to the parameters of interest. And Frank has introduced this term marginalization, of trying to get information about the parameters of interest as opposed to the nuisance parameters. Okay, so here's Frank. He wrote a pair of papers with his student Brad Treese on 2019 and 2020. And this really says, for a reflectivity curve, how do we quantify the amount of information we have? And we're going to do that in our parameter space. Remember, we have a model. It's parameterized by these hopefully physically meaningful parameters. And as we do the measurement, those parameters are constrained to smaller and smaller confidence intervals. If you have a Gaussian and it has a width sigma, you can calculate what the information gain from a uniform prior. At the beginning, you say, I've got some range, I don't know anything in it, that's my uniform prior. And relative to that, you can calculate what the information gain is for a single parameter. So as your width of your distribution, as you learn more about your parameter, the width of the distribution, the posterior density function, or typically think of it just as the standard deviation maybe, gets smaller, and the amount of information goes up. You can extend this calculation to multiple parameters. And in this particular case, this is called the multivariate normal approximation. You would just take the covariance matrix, and it's the negative logarithm of the determinant of the covariance matrix. And the reason that you have to have this is, imagine you have two parameters that are equal to each other, A equals B, right? If you get some information about A, you automatically get information about B. And so you need to have the cross correlation term, which is what the covariance matrix gives you, to make sure you don't overcount the amount of information you've got. There are other ways of doing this calculation that are more accurate. There's a Gaussian mixture model, for example, and some weighted mean distances and those sorts of things. And which one you choose may be related to how much computational power you have and what the needs of the measurement are. For everything I'm going to show here, it's all in this multivariate normal framework. The other advantage of this is that for marginalization, you just choose a subset of the parameters you want. So I had 16 parameters in my model. I'm just going to choose 5 of them, isolate the rows and columns of the covariance matrix for those parameters, and calculate that as the information, the marginalized information in my measurement. Okay, so that's a sort of quick and dirty way of doing the marginalization. All right, once we're armed with these concepts, we can actually come up with a strategy for doing our active learning. And now remember that what we want to do is we want to optimize the rate of information flow into my experiment per measurement time. So this is going to be the marginalized information that I've gotten divided by the amount of time it took me to get there for each step. So this is my rate of information. And I'm going to just call this the figure of merit at each measurement point. And the idea here, here's the general strategy, is to, at every point, you have some data that you've already taken. You then search through all the possible instrument configurations, X. In this particular case, this is just going to be Q for a reflectometry measurement. So this is X or Q. You use your previously collected data to produce a best guess for the count rate at each X. And then from this, you determine the expected rate of information increase from that point at each new data point. So that's the figure of merit. So you measure a data point, you fit the data, you recalculate the figure of merit and identify the next data point, and then you repeat over and over again. So what does that look like in practice? Well, the top graph here is showing reflectivity as a function of Q, and this is for a completely unconstrained model. This is actually for the model that I showed you previously. And and what you're seeing here, each one of these curves in the spaghetti is a single random draw from the prior distribution. So basically, I just take some random set of parameters in parameter space, calculate what the R of Q curve would be for that, and plot it. And you do that over and over again. And so this is basically your space of possible R of Q curves, completely unconstrained because I have no data. Nonetheless, I can still calculate a figure of merit, and it says you should, and I'll talk about what that is. And it says you should measure here. So you take a measurement there, then you recalculate the figure of merit. You shouldn't measure there again. You can see that it's strongly suppressed now because you already have data there. It says you should go measure over here. You go measure over there. And again, now you see that all the model curves go through this data point now that I've measured because I'm constraining the model. And now I have a new figure of merit and a new next point. So we're going to do that over and over again. So how does this work in practice? So in practice, what we're doing is when we do the analysis at every data point, we come up with a new distribution, a so-called posterior distribution function of the parameters. And so basically, this is the space of the parameter space that's allowed by the data that I've taken so far. To quantify that, the probability of having a particular set of parameters X is given by the log likelihood function here. It's exponential in the negative chi-squared over 2. Now, one thing to remember here is that each time we sample this distribution, we get a new set of parameters that could describe our model, that's allowed by the data we've taken so far to describe our model. And each one of these corresponds to a possible R of Q curve. So in this previous graph, each one of these curves that you see here corresponds to one parameter set xi. So just as a sort of visual example, this isn't a real example, it's just a schematic with 2 parameters. You've got parameter 1, parameter 2, you've taken some data, and they are constrained to have this relationship with each other. This is basically the cross correlation between parameter 1 and parameter 2. Each one of these dots is a different set of parameters, parameter 1, parameter 2, and each one of them corresponds to one of these R of Q curves, of which I've only plotted 3. And then the black curve here is the so-called ground truth. It's the thing I should be converging on. The way that this information maximization algorithm that we've developed works is as follows. For each Q value, you go to that Q value and you take a slice through the R of Q curves that you've calculated for all your parameters that you sampled at each one of these Q positions. You plot those as a distribution. You don't have to plot them, but you can think about this as doing the following. You plot them, and let's say it's a Gaussian distribution for the sake of argument. So this is the probability that you get R as a function of R, which is basically a slice, a projection on this plane here. Then you say, let's say I were to measure at this point. My best guess for where I'm going to measure is at the median of this distribution. And if I measure for long enough, I will have error bars on this data point that are tight enough that, and if I measure at the median, that's my assumption, that the tails of the distribution will no longer be allowed. Which samples correspond to the tails of this distribution? Remember, each one of these curves corresponds to one of these sample sets. So I can identify which of the samples are in this red region and which are not. So I've color-coded the red ones in red here, and the blue ones are the ones that would be excluded if I were to measure here at the median. So I look at this and I say, well, look, my red points and my blue points are evenly distributed throughout my parameter distribution here. So I won't actually have learned anything about my parameter space here if I do this measurement, because I've evenly excluded samples throughout the distribution. By contrast, if I were to measure somewhere else, let's say here, and I were to do the exact same procedure, maybe I would find that the tails of the distribution all correspond to this cluster of parameters, in which case I've learned a lot about my parameter space, because now I can exclude this entire cluster of parameters. Both parameter 1 and parameter 2 are going to have a smaller variance than they had before. And this is a key point. You're basically trying to find measurement points that are going to reduce the variance of your parameters of interest. Okay. So in this particular case, you'd have a low increase in information because you haven't decreased the variance of your parameters. And in this case, you would have a high information increase. But now, what does it cost you to get there? It takes you some time to measure. And actually, for a scattering experiment, this is great because we know that our measurement time obeys Poisson statistics, so we can calculate exactly how long it's going to take us to get there. If you have a point like this one, which has a broad distribution in R of Q, the R of Qs are well separated. You only have to measure to an error bar, a relatively loose error bar. It doesn't take you that long to get there. And you can calculate how long it is just from Poisson statistics, which is here. Here, sigma 0 is the width that exists in the distribution. Sigma is the width of the distribution that you want to get, and R is your counting rate, which you assume to be the median. This one, on the other hand, the R of Q curves are all very tight. They all sort of cross at this region. So this is a rather tight P of R distribution. It's going to take you a long time to get there. In other words, even though you get a lot of information from this measurement, it takes you a long time to get there. And this may not be the optimal one. In the end, what you want to do is you want to optimize the rate. So you want to find perhaps an intermediate information gain, but one that doesn't take you much time to get there, for example, may be your best option. But this is all done automatically. It's done point by point. You go through all the Q values, you do this calculation, and then you calculate the figure of merit. And what does that figure of merit look like? Okay, well, this is an example of a figure of merit for an experiment. So just look at the orange curve here. You've gone through Q, and this is your figure of merit. This is your delta H over delta T, and it has some points that are better than others, and then there's a clear maximum. Right? This is where you would take your next measurement. Okay. And so how good is this? Well, look at the maximum of this orange curve relative to its average. Right? It's probably about a factor of 2 or so better than the average. And that gives you some sense for how much better you're doing. This particular analysis also allows you to do something clever, which is you can not only look one step into the future, but you can actually look multiple steps into the future. You can say, well, now I'm going to take the I'm pretty sure I'm going to get these red samples when I'm done. So now I can do the same thing again, and I can take another subset of those, and you can go a few steps into the future. And for things like X-rays, this could be helpful because the analysis might take longer than the measurement does. So having a few points in your queue could be useful. You can also incorporate other costs, not just measurement time, for example, movement costs. For our monochromatic reflectometers, it takes maybe a minute to move from the maximum Q value to the minimum Q value. And so this can actually affect where you might want to go. So here's an example where the red curve shows the figure of merit for some step. I'll just call it step 0. There's a clear maximum out here at high Q. So you measure there, and then you recalculate the figure of merit. You don't want to measure there again because you just took data there. So you want to measure somewhere else. And you can see that in the green curve. But now if you put a movement penalty in, normally you'd want to go all the way here, which is the maximum of the green curve. But it actually costs you some time to move there. So actually, once you put the movement penalty in, you find that this is actually your optimal measurement point. So you can you're slightly more likely to measure nearby points if you put in movement time cost. So all this can be included in the algorithm. Is basically the point. All right, so then you just cycle through this over and over again. Now I'm just going to show this little video here of how that works. In the top left, I'm showing the data, and the spaghetti again is are the possible RFQ values that are sampled from the posterior distribution. Sorry, they're the RFQ values that are allowed essentially from the data you've already collected. And what that means is if you sample from the posterior distribution randomly, those are the RFQ values that you get. The figure of merit is shown here on the bottom for each one of these steps. And you'll notice a couple of things right away. There are some places where it measures a lot and some places where it doesn't. So for example, out here, where you have this minimum of the reflectivity, there are very few measurements. That's mainly because the intensity is low and the signal to noise is low there, which is built into the algorithm. But there are other places like here around Q of 0.1, where the model is actually so well constrained by the data around that point that there's actually no reason to measure there. All right, and this is one of the advantages of having this model-based approach. Okay, and as time goes on, you get more and more information. It gets better and better constrained. What I'm showing on the right is the information gain as a function of log time. And the blue curve shows the autonomous measurement, and the black curve is a control measurement that I'll discuss in a few minutes. The top shows the marginalized information, so about our parameters of interest, and the bottom shows the total information in the measurement as a function of time. If there are any questions, this might actually be a good place to take them, just for on this part of it. If not, then I'll keep going, okay?
No, I do have one.
Yeah,
Sorry.
go for it. Yeah.
So when you say you want to constrain the parameters for this kind of things, so how do you constrain them basically?
By taking data, right? So the idea here is that, yeah, this is a key point. Let's go back up to this slide, because this is really the sort of the key thing to understand. All the data you've taken so far give you this population of parameters, okay? Yeah. Parameter 1 and parameter 2. The idea is to say, by measuring, I'm going to change this distribution, okay? If I take another data point, this distribution is going to change. But I want to change it in a way that reduces the variance of the parameters I'm interested in. So this whole process is just identifying which data points do a better job of reducing the variance of the parameters you're interested in. Okay, does that make sense? Okay.
David, I have a question also about this data taking. I'm very ignorant and I'm neutron scattering. So in your measurement, the whole measurement, you don't need to attenuate the intensity for the detector. Is that right? The whole intensity range fits your dynamic range of detectors.
That's correct. That is correct. Now, though I would say that if you did need to attenuate it, usually you would take that into account in your data reduction process, right? So you would know that you put an attenuator or not in. But for example, if it took you time to put an attenuator in, I assume this is done automatically, then you could actually take that time. You could say, oh, if I go and measure here, I'm going to have to put an attenuator in. That's going to take me 30 seconds, right? So you could include that as a cost to measuring at that particular point, for example. But yeah, in this particular case, we don't have to attenuate.
Okay.
We do attenuate when we take our direct beam scans, of course, but the reflectivities are so low that we don't need to.
Okay.
I also have a question, if possible.
Sure.
The way you take the points are jumping from point to point. Now, suppose what happens in X-rays, not in neutrons, that the beam drops. So you end up having a partial reflectivity curve with some points here and some points there. If you were to measure sequentially, you would have gotten a full reflectivity curve, although not optimized for intensity. Am I correct?
Yeah, so that's right. So if you if your beam goes down, I mean, if you're measuring point by point, of course, it could go down, and then you'd still get a partial curve. If you were concerned about that, again, you could, for example, imagine altering the algorithm to include as part of the figure of merit how much space you have between points. So it would basically say, you could say, look, I want to make sure I map out the whole curve. So I'm going to measure at my optimal points. Unless there's a huge gap between some data, and then I'll measure in the gap. Or, for example, you could take a low statistic scan through your whole curve, so that you mapped out the whole thing. You don't have any surprises, and then turn on the autonomous measurement. Right, so you can kind of see the whole thing, and it will actually have a good starting point because you will have measured something about the whole curve, and then turn on the autonomous measurement. And in fact, when I was first doing these simulations, that was actually what I did. I actually I did it that way. Turns out you don't have to, but you can.
Okay, thanks.
Yeah.
I have a question and a comment. I think this is beautiful because what you're doing is basically concentrating on as you go, basically. I guess you're doing fitting while measuring, if you understand you correctly. Yeah,
that's correct. That's correct. Yeah.
Then, but one of the problems that we have usually is that some of the parameters are correlated. So how do you overcome that?
So the
yeah, I mean, I guess you're still using what we call the box model.
In this particular case, no, this is one of these composition space models, but you're right that the parameters are strongly correlated. So for example, two of the parameters in this problem are the thickness of the inner and outer leaflet of the lipids. We don't actually have good contrast between these. So these are strongly correlated parameters. The total thickness is a good parameter, but the individual leaflet thicknesses, you have to measure for a long time to really constrain those. So
what is the gain in information? You're showing us the reflectivity, but you don't show us the electron density. How much information do I gain if I do that? And is it worth it? I mean, you always,
right.
It's always a balance of how much time you're going to dedicate to something for the reward of it. Yeah.
So the correlations between the parameters are taken into account in the information measure. And the one way to see this is that imagine you had two parameters in your model that, you just threw an extra parameter in there and said, okay, parameter A equals parameter B. There are two parameters that are absolutely equal, always. These are two strongly correlated parameters. They have a correlation coefficient of 1. So what happens in this particular case is the total information that you calculate with this MVN calculation, for example, takes into account the off-diagonal terms in the covariance matrix. And so you would never see twice the information gain from parameters A and B, which are the same, as for just A. It's all taken into account. All comes out in the wash in the information calculation. Hopefully that answers your question, but it's all taken into account. As for the question of how much time do you spend on it and that sort of thing, I'll get into that in a couple of slides. Okay.
David,
yeah.
sorry, I have one more
No, go
question.
ahead, please.
So it's just by looking at this movie, it seems like you are trying to count the same point over and over again. Maybe, if you swing, back and forth a little bit, and most likely you skip all these minimum points. And why they always keep coming back to the same region, It's just because it didn't count long enough?
That's right. Right. So the algorithm basically, again, I'll talk about this in a couple of slides, but just to give you a preview, what the algorithm does is it says, I'm going to measure the length of time I'm going to measure is going to be related to how much variance is in the reflectivity at the point I'm measuring. So if there's a lot of spread here, it's not going to measure for as long because to constrain that distribution doesn't take as much time. Whereas if it's a tighter distribution, it decides to measure there, it's going to measure longer because you want to gain information about, or you want to reduce the width of this distribution. So so basically, there's a sort of a tuning parameter in here, which is this eta, which is how much of this distribution do you actually want to exclude. So you could be super aggressive, and you could measure here, and you could say, I want only the sort of really inner band of this. So I want to exclude 90% of the samples. This would be a really aggressive measurement. You can also be really conservative. You can say, oh, I only want to exclude just a few samples on the edges of the distribution. So this is a sort of tuning parameter of the algorithm, and I'll talk about how that plays out in a bit. But yes, what will happen is and the reason you don't want to be too aggressive is because that may not, especially early on in the measurement where you actually haven't really learned the structure of the problem yet, that may not be your best place to measure. And so you want to spend a small amount of time there, right? And then return to it if later, after you know more, that actually ends up being a better parameter. It's actually all, the algorithm just kind of does this though all on its own. But you're right that you are going back and measuring over and over again, sometimes in the same point. So that gives you additional movement times. But in this particular case, so I actually keep track here of how much is movement time and how much is measurement time. And if we let this play out, I wish I could fast forward it. It's I should do an MP3 or MP4 instead of a GIF, so I could fast forward it. But you can see that early on, the movement time is a big fraction of the measurement time. But as you go on further in the experiment, the measurement times get longer and longer and longer. Basically, these points are almost evenly spaced on this log scale. And as you do that, the amount of time you spend moving becomes really small again. And that's after you've learned. So basically, when your measurement times get long, it's after you've already learned a lot about the structure of the problem. And so then you're really spending all your measurement time on the optimal points. So let's actually talk about how well we do. So how do you actually measure the performance of an algorithm like this? Because you want to make sure you're doing better than your typical measurement would be. And so the naive way of measuring a reflectivity curve would be to just say, well, it drops off as q to the fourth. I open my collimation, so I get a q squared back. So I'm going to measure my time at each point as a function of q is going to be q squared. And if you naively do this, and you run your autonomous measurement, that's the blue curve here, then your control measurement is the black curve. And what each point here means is I have some scheme that I usually use for taking data. I say I have a total time here of say 10 to the 3 seconds, and I'm going to distribute that time among those points according to this weighting factor. And if you do that, so then you simulate data with that total time with that scheme, and you look at it relative to the autonomous scheme. If you do that, you can calculate the speed up factor, the acceleration factor, how much better you do, which is basically the horizontal distance between the blue and the black curves. And you could do better by about a factor of 6 in your measurement speed. In other words, to get to the same level of information in the measurement, you get there a factor of 6 faster in time. Now, of course, we don't actually take data here like this. We have, some decades of experience on samples like this, and our current best practice is actually this particular equation. This is the weighting factors proportional to this. And if you do that, you don't do quite as well, but you still do about 50% to a factor of 2 better. And you do even better than that at short times. You could also say, well, what if I were to just calculate I have the figure of merit. What if I calculate the maximum value of the figure of merit, which is my information rate, relative to the average? So this is basically if I were to, at every point, just randomly select a point to measure at, how much better do I do? And it's about a factor of 4. So somewhere in here is basically your typical speed up for this particular sample. And what I want to point out is that even though we have decades of experience, we do pretty well with our current best practices, but the autonomous measurement actually does better than that. So really, this sort of sets the standard for measurement efficiency. And I don't want to imply here that this algorithm is necessarily the best one. I think the search for a perfect algorithm is something that people are always doing, but this one seems to work pretty well. It also shows that we do pretty well with our organic intelligence, our experience, our intuition for common samples. I'll show you other examples in a few minutes where we do better than that. So this goes back a little bit to a question I got earlier about how do you know when it's worth it? So this shows, because you're analyzing your data constantly as a function of time and as a function of steps that you take, you can actually track the errors in your parameters as a function of time. So these are the 16 parameters in the problem. I've highlighted in red the 5 parameters that I'm interested in, so the chain thicknesses, the submembrane thickness, and so on. And you can actually see how they change in time, and the black is the control measurement. Our best practices control measurement. So you could actually say, look, I want to know the submembrane thickness to 1 angstrom. And if you wanted to know it to 1 angstrom, you could have stopped after 400 seconds here, and it would have taken you, 1,200 seconds to get there with the with your control measurement, for example. So that would be an example. So you have real-time decision making. This really sort of empowers your users to stop when they're done collecting data. I also want to talk a little bit about this parameter selection. So here's an example where I ran the exact same simulation, and instead of selecting those 5 parameters about the bilayer that I was talking about before, what I did instead is I selected the intensity parameter, which is just a scaling factor on the intensity of the entire curve. And so this is a completely different parameter. And what you notice is that you still actually map out the curve, but what it's not doing is it's not spending very much time on these measurements. It's actually spending all of the time down here at low Q, where you get a lot of information about the intensity. So you see these big jumps in time. Each one of those is a very long measurement at one of these low Q values. Okay. However, it does sort of search the rest of the Q space just in case, I suppose, there's information out there that it needs to learn that, whether or not there's information out there. If you look at the parameter errors as a function of time, I just want you to focus on this little plot here, which is the error bar on the intensity as a function of time. The orange points are the points I showed you before, where we were selecting for the bilayer parameters. The black curve is the control measurement, and the blue curve is where we're selecting for the intensity. So it speeds up the information you gain about this intensity parameter by about an order of magnitude over when you're selecting for the bilayer parameters. This kind of shows that the parameter selection works, at least in this particular case. You can also compare the measurement time histogram. So this is how much time it's spending as a function of Q for the 2 cases. And in the case of the bilayer parameters, it spends a lot of time out here in this intermediate Q range, which is giving us a lot of information about the bilayer structure. For the intensity parameter, it spends all of its time at low Q, where it knows the most about the intensity, and only a little bit of time at higher Q. So that's sort of working. Now, one of the things I said at the beginning is that with reflectivity data, we often collect complementary data sets, and this basically helps us make up for the lack of brightness in our sources. In this particular case, this on the left here, I'm showing a model corresponding to the same sample in D2O and in H2O. And so in this particular case, what I've done is I've just allowed it to switch back and forth between D2O and H2O without any sort of cost. This is obviously not true in reality. You'd actually have to exchange the fluid in the liquid cell that we use for this to do this. But this is informative because it shows that it gets information from both the D2O and the H2O, but you can also kind of look at how long it spends on each one of these models, and actually spends about twice as much time in the D2O model as it does in the H2O model. And if you look at some of Frank's work previously on experiment optimization, he finds that this is actually a pretty reasonable ratio for most of these types of samples as well. So we can sort of predict what the optimal time perhaps that you might spend on D2O versus H2O contrast to get the most information about your sample. For this 2-model system, the speed up that we get relative to our best practices is actually even better than it is for this single D2O only case that I showed you before. I pointed out that you could forecast several points into the future. You can go about 5 points into the future before you actually start to degrade. And what I mean by degrading your performance is your speed up starts going down. The marginalized entropy as a function of time starts to move toward your best practice or your control curve. So you don't see any degradation of performance if you're forecasting about 5 points into the future. So this is useful for instrument control, because if your analysis takes a long time, you never have to wait for it. You can keep taking data even while you're analyzing. Okay, and then this is this aggressiveness parameter that I was telling you about. So there is this tuning parameter in the algorithm that says, how aggressively do I want to take data? The more conservative you are, basically, the fewer samples the less time you spend measuring at each point, and you measure more points. So that's cost you a lot of computation time, but if you're not worried about that, it's okay. If you get, so 50% here is basically saying, I want to exclude anything outside the 50% confidence interval of my distribution, and you get some performance based on that. You get some speed up, again here about a factor of 2.5. If you say instead I want to exclude 70% of my samples, you see a small degradation of your performance. And by the time you go really aggressive, you say I want to exclude 90%. You're basically overcounting at each point. And the problem with that is you don't know yet that that's a good point, and so you end up getting a real degradation in performance. In fact, you can even do worse than your sort of best practice. So it's not very sensitive to reasonable values of this parameter, but it's something to keep in mind. There is a tuning parameter in the algorithm. We have a new reflectometer, which is called Candor. I'm not going to spend a lot of time on this because I'm at about 45 minutes already. But this is a polychromatic reflectometer. We can measure multiple points at once. And basically, what this means is you're just smearing out the figure of merit. So you don't get as sharply peaked points, but you can still do the same algorithm, essentially. And so you can see the data are coming in chunks. Let me see if I can actually restart this. Yeah, here you go. So data are coming in chunks. In Q space in the polychromatic reflectometer, and you can still do the same thing. And in fact, this is a brand new instrument. It came online right before the reactor went down. We got about one cycle of data taking on it before our reactor went down a couple of years ago. And so we're really looking forward to trying this out. We didn't actually have best practices for this instrument. And so if we look at our performance, we actually do really well relative to what we were doing before, about a factor of 4 in speed up. However, if you look at the performance relative to this randomly selected point in the figure of merit, you only get about a factor of 2 compared to about a factor of 4 for the monochromatic reflectometers. And this has to do with the fact that even if you're measuring at your optimal point, you're also measuring some points around that, to the sides of that may not be quite as optimal. So it's not, you don't expect it to do quite as well for a polychromatic instrument as for a monochromatic one. Here's a different model. I can't tell you what this is. This is like this isn't published or anything yet. But basically, you can see the difference in the scattering length density profiles that we're trying to, or the type of thing we're trying to look at in the scattering length density. It's got this sort of interfacial, some stuff piled up at an interface essentially. And in this particular case, we don't have best practices for this because we've never measured this type of sample before. I just ran it through the simulator, and the autonomous measurement does over an order of magnitude better in terms of time. And the reason for this turns out to be that all the information that you want is concentrated in a fairly narrow region at low Q here. So all the time you would spend out here at high Q is basically wasted. And you know, for this, you should focus on the low Q measurements. And the model just picks this up. The simulation just, you know, finds this automatically, and you're off to the races. So what is this good for? I think for an audience of beamline scientists and reflectivity experts, hopefully this will be clear, but there's kind of 2 use cases that I can think of where you might use this move going forward. One of them is just to find an optimal distribution of measurement times in Q space. You can, the signal to background ratio is something that you put into the model in the beginning. You know what your background is typically, and so you can add that. And so this shows on Candor, I let the maximum Q value, this is tiny for you guys, but it's huge for us, go out to a Q of about 0.5, and then looked at how much time a Candor spent at any one of these Q values, remembering again that the signal to noise ratio out here is really poor. And what we found is that it spent most of its time in this sort of intermediate Q region, and so you could actually then say, okay, I'm going to modify my best practices to reflect what the simulation has shown. Additionally, for interdependent models like this H2O D2O, you know, simultaneous fitting, you can get optimal distributions of counting times from that. I'm going to skip that. The second use case is instrument control. Of course, you get faster data collection times, which at a intensity limited case like neutrons is just hugely important to us. Even factors of 50% or a factor of 2 is just enormous for us. You also get some real-time user decision making. You can monitor your parameter variances as a function of time. If your sample ends up not being what you think it is, you discover that very early, typically in this process, and you could try to change your model mid-experiment. And for neutrons, again, we're statistics limited. Systematics aren't usually a problem for us. at least for shorter experiments. And so we can actually monitor our fit quality for the onset of these systematics or model fitting issues. At some point, systematic errors dominate, and for the way that we usually treat our data, at that point, collecting more data, we're not often going to learn a lot from that. And then finally, from a sort of user care perspective, our experiment ends with already analyzed data, so they can be sent home with data that are essentially already analyzed, assuming everything goes well. The sort of open questions, I think, about this that I'm interested in exploring going forward. There are a few next steps. I mean, for the neutrons, polarized beams are important. What happens if you have some derived parameters? All things that we can talk about, but I really want to focus on the second one. What if the model's wrong? Right, so what if you actually don't have a good model for your system? How robust is the algorithm? How wrong can it be? How bad can your systematic errors be before this starts to fail? Or alternatively, what if we don't have a model? And there's a whole series of there's a whole effort in this autonomous learning community, especially using things called Gaussian processes that are a promising candidate for taking data when you actually don't know what you're going to be looking at. Right now, this is out on GitHub. It's not particularly well documented, to be frank, but basically there's this, you can run this reflectivity experiment with a REFL1D fit problem if you use REFL1D, and then it'll keep track of each step in the experiment and collect some simulated data that you can then use to analyze later. If you're at all interested in this, by all means, just feel free to reach out to me. And that's basically it. So just to summarize, what I've sort of demonstrated here is that with these simulated data is that you can calculate this real-time figure of merit from this Bayesian inference process, in this case, a Markov chain Monte Carlo sampling, that allows you to identify the next data point that's going to give you that, based on what you've done so far, you expect is going to give you the best information. It doesn't have to be perfect, it just has to be good. You get at least a factor of 2 speed up over current best practices, and it can be much higher than this, especially if you don't have a lot of institutional knowledge about your samples. There's a lot more to be done. And then there are a couple of surprises here. Remember I said at the beginning that our central assumption is that some regions of Q space are more efficient than others at constraining the parameters of interest. And of course, we're talking about the low signal to noise or below the critical edge, but also inside the sort of the information rich Q region. There are also some regions of Q space that are more efficient than others at constraining its parameters of interest. And it's not at all obvious that this is going to be true, right? We're taking real space parameters, we're doing a big Fourier transform on them when we scatter, we're spreading that information out in Fourier space. It's not at all obvious that some of those Fourier components are going to be much more efficient than others. And that actually turns out to be the case. So that's kind of cool. You can also do this for polychromatic instruments, which I wasn't sure was going to happen here. So I think that's the end of my talk. Thank you for your attention. Thank you for all the good questions so far, and I'm happy to take any other ones.